By using readily available cheap carboxylic acids and activated selective CH bonds, you could synthesize this molecule in just 2 steps. So that is one of the very powerful techniques of doing selective CH activation. So one of the molecules that Pfizer developed was this lead hit molecule, which is basically M1 positive allosteric modulator, that is muscarinic, usually basically to regulate your Alzheimer's and Alzheimer's disease and schizophrenia, people suffering from schizophrenia and Alzheimer's. This is the drug which is given to moderate them, to give them stability. So this was created based on CH activation from simple benzoic acids. Okay, so this is one of the this shows you the importance of the CH activation. Now, what have we done? What have we done let's go back a little bit into the histories. So, in 1950s, we were just doing functional group transformations, and in 70s, we started using transition metals for coupling reactions and so on. In 1990s, we came into catalytic CH activation. That is what it came into. So, this is going a little back into the history. Sorry. Organic chemistry, 50 years of modern medicinal chemistry, dormant in CH bonds, but then, now we have this powerful tool where many molecules, drug molecules, can be synthesized in fewer number of steps. Okay, so going further from here, this is the previous report of the work. Okay, just to give you a little bit of background. Now, CH activation is not possible just like that. Okay, for the CH activation to take place, you need a very good directing group. Okay, what DG stands here is for the directing group. So, you need a very good directing group where the metal comes and coordinates, abstracts the proton, and activates the adjacent carbon. Okay, so that's why you need a very good directing group. And Professor Hugh for CH activation, like CH amynation and olefination and so on. So where is the gap here now? We also know that alkenes and unactivated alkenes are very difficult to react because they are unreactive. The same as your CH bond, they are unreactive. So and also with getting stereospecific regiodivergent selective reaction is very difficult, and is less explored. So how do you get this regiospecific by using the unactivated alkenes using one component catalytic system? Okay, so that is what we found the gap over there. different like methylamine or N-methylamine and arylamines and all, and the halogen substituted arylamines. And we also looked into some poly. This monosubstituted fluoroaniline was gave the best result of 73% in basically 20:1 ratio, like 20 as your major product is your branched and the linear as the minor product. So this is what is the best. We also use some bidendant ligands as well, but the reactivity fell. There was not much great product. Okay, so we were able to perform this reaction in gradient state, that is 10 mole%, and monoamino protected amino acids, that is N-glycine, N-acetylglycine, and then your DCE and at 80 degrees and at 14 hours we were able to get this product. So going further, screening of ligands, we took different ligands, like as I said, we took different amino acids, sorry.
F25, F5.
Sorry?
Okay. Yes, okay.
So we had this. Okay, we took different ligand screenings. So we took different amino acid, protected amino acid, acetyl protected amino acids, and the best we got is we also took some bidendant ligands and the phosphorus ligands. The best we got was the results with N-acetylglycine. That is the best results what we got the ligand. And where we got the branch chain, in case of styrene, we got the branch chain as the major product. Now, scope of unactivated aliphatic olefins, like you have unactivated olefins. So here what we got is basically the major was in the presence of your palladium state and so on. We got the major product as the branch chain. So these were the different substrate scopes that we used, different kinds of unactivated olefins, and most of the major product was your branch. But when we took the cyclic styrene, when we took the cyclic styrenes, we got exclusively branched. We did not get the minor product as the linear as the minor product. Okay. So going a little bit quickly in this aspect, so we also next we took is basically we also developed the crystal structure of this these two molecules, and we were able to see that the major product was the branched product. So going further from here, the substrate scope with styrene, when we took the styrene, the major product was the linear product. We did not get the branch product. Major product was the linear product, as you see over here. As Professor Verma also said now, that deteriorated compounds are becoming more and more important in drug industries basically, and they are very expensive also. And with respect to medicinal chemistry also, deteriorated drug molecules are showing some enhanced potency than the normal molecules. So we also did this reaction where we took where we did the deterioration of using D2O in DCE and palladium on same reaction condition, but we used D2 and we were able to substitute the deteriorated products in quite a good yield. Yes. So further mechanistic studies was done where I think you explained it in much detail using the real-time mass spectrometry, but we don't have that facility, so we were able to first we had to synthesize the complex, that is with your palladium and monoprotected amino acid. We synthesized the complex, and then we ran the LCMS. We do LCMS, and then we were able to see that the different the acetonitride. Okay, and we also saw another thing is substrate with the palladium and the monoprotected amino acid and styrene. So we get the respective isotope molecular mass, which confirmed the mechanistic study, which basically, okay. So going further from here, mechanistic studies, as we said, we also used different rich and electron donating groups on the substrate here, CF3 and methyl. With slight changes, we saw that they gave majorly the branched product was the major product. Further intermolecular this one with competition, like in a competitive reaction, we also saw that by using this 1:1 of your substrated and deuterated substrate with the styrenes, what we got is KH by KD was 2.3, indicating that the formation of more of a branch chain than that of the linear chain. Okay, so we further did these reactions, and in case of styrene, we got the linear chain. Okay, so because the KH by KD was a kinetic study was basically 2.3. Okay, if it is greater than 1, then it is basically favoring the CH activation. This also proved that it was not a normal electrophilic substitution reaction, but rather the CH activation was a rate determining step in this process. Okay. So proposed mechanism was the proposed mechanism. I will not go much in detail, but as you can see is the two major intermediates over here, which leads to the beta hadad elimination, and you get basically these linear chain and one favoring to the branch chain in this case. So this is, I'm sorry, for some reason this one is not very clear. Otherwise, so in calculations, we did some DFT calculations. As you will see that in the path, you'll see that the transition state for the formation of the branch product was more favored. Especially if you look here as a 15.7 as a transition, here 25. These are the major two blocks where the formation of it assisted in the formation of your branched chain rather than linear. So in case of the styrene. So in case of styrene, the formation was mostly linear. Formation was more favored to the linear, and this is what it showed here. The transition, this 1 kilocalories was the transition state difference. So we also perform gram scale synthesis, and also one of the important thing is how to cleave the amide bond. So that is also we did. We were able to achieve that, and we were able to scale up the reactions as well. So it can be implemented in large scale synthesis, as well as we were able to cleave the amide bond by using some very normal basic reactions, and which can further be used for derivatization on this side. So advantages of our method: CH activation with unactivated aliphatic olefins is still underdeveloped due to low reactivity. Only few groups in the world have reported this branch selectivity of CH proliferation with unactivated aliphatic olefins, and we are one among them. Regio-divergent olefination with single catalytic cycle is underdeveloped, and we have achieved it. And then further CH deuteration with our optimized reaction conditions, since deuterium plays a crucial role, in drug discovery. Useful of readily available and easily removable monodentate directing groups. And then further application of synthesized derivatives, gram scale synthesis. So these are some of the advantages. Challenges what we faced, these were challenges, how to read it, but some of these were maintaining the mild reaction conditions, achieving regio-divergency with a single catalytic cycle. So in conclusion, this is what it is one of the very well-known reported ways of olefination of unactivated alkenes in pyramid. Maybe this can also be applied in developing other scaffolds as well. Further, this was accepted in Chemistry European Journal as one of the hot papers, like it was a hot paper of that issue. Further to this, we continued with our work, and this is our latest addition to that. We just recently got accepted where we have used no ligand here. We have just used ethanol and so on. So this is, I'll not go much in detail in this. So no ligand, and it was only ethanol and water, and oxygen as the oxidant, and we have also done mechanistic. Basically, we have replicated the same work, but without any ligands. Okay. Now going here, this was accepted recently, just a month back, and future goal is to have towards sustainable CH activation. That is one of the things. This has given us an interest in developing more CH activation mechanisms and working with CH activations. We have achieved some more interesting work in this field, which we have just communicated with the green chemistry, and probably should be out soon. So just a brief, my work with mechanistic studies started in 2015. My collaborator Milan Beran, and he was my first PhD student, Stuart Prananidhi. This was a sad start because the moment we got this work done, it was published in the same week when we wanted into organic letters. So we had to submit it a very low, this one. I'm sorry about this. It's my daughter's picking up from the school. So that's what is that. Okay, this is what, and I'll go quickly from here. So we did some more work in copper catalytic terminal reaction. And each of these reactions are not towards just doing a chemistry, but towards building a new scaffold, from where I have developed some very potent anti-malarials, anti-tubercular, anti-cancer agents, lead molecules. Okay, so in this quinolone kind of moieties, quinolones, we have got a series. I'll show you in Team to Drug Discovery. We've got some very interesting molecules. Going further like this, we have also done this was done by Naveshwar. So here also we use some very cheap starting materials to use benzoxazines, and benzoxazines are core scaffolds of most of your amoxicillin, ciprofloxacin, ofloxacin and all. Okay, and I work on DNA gyrase, and these work on all amoxicillin, oxacillin are basically DNA gyrase inhibitors. Okay, going further, just quickly, so we also did some work on transamidation work. Very recently, we published some transamidation work where the breaking the amide bond is also very difficult and doing some substitution, so we did this kind of work. Then we did some DBU reactions here, triazole-triazines, and these have shown as excellent anti-malarial activity molecules, which we have built. Okay. Further going from here, as I said, I'm a medicinal chemist. So you can say one of our highlight was this paper in 2016, where we have achieved 0.48 micromolar against INHA resistance, fluoroquinolones resistant strains, MDR strain, XDR strain, and so on. Because my group has a facility towards BSL-3 facility we have, where we can do the screening of all this. We have a facility called all tissue cultures, and also have built that kind of thing. So we have done a lot of work, and you can say that we have achieved, in most of these cases, we have achieved some excellent MIC values. Not everything translated into patent molecules, and not everything translated into a drug, but some of them we were able to further patent these molecules. Similarly, antimicrobials, we did work with bromopirols and other molecules and also antifungal agents. This is some of our work that I'm just showcasing the people who did the work. So this is one of our lead molecules which has shown what is for MRS they use the last resort drug is vancomycin, am I right? And vancomycin, our molecule is equipotent to vancomycin. So this is what we are, the unpublished work, and we are in the process of further investigating, doing toxicology studies and animal studies. And this work was done by Meridu, Srinivas Meridu, and believe me, some of the PhD stories are not good. because until the end of this final year, we were unable to publish anything. And finally, we took the molecules, and always believe this, there is, you do the greatest discovery out of serendipity. Serendipitously. You never know, you're doing some reactions and all. All the professors over here will agree that when you do a reaction, or in any case, never throw away, investigate each and every isolation what you do. You may come across some new things, or new chemistry. So that is what we got in this case. In the end of the thing, we just screened it against, the molecules were built for tuberculosis, but end of the time we got it against high potency in MRSA. Sorry. So these are, in 10 years time, we were able to have this anti-TB targets. So we patented, we patented these molecules over here as anti-TB targets. We have achieved 1.9 to 0.78 micromolar. This is the quinozole, as I said, within the methodology, quinozole known. So we were able to get the entire series as low as 0.3 to 3.25 micromolar. The entire, like 30 derivatives were all active. So we are busy now doing investigations. This is the dihydrogingerone semi-synthetic compound. So going further, just to conclusion, we also work, as I said, Francis joined me from UK, and he wanted to work on malaria, so we did some, and we published some very interesting results, and yet we have just submitted another we have achieved anti-malarial parasitic against the resistant strain at micro and nanomolar concentrations. Okay, so that is what we are at. This is one of the very interesting work that I did with UCL, with Frank. He works on spindle kinase inhibitors. Now, spindle kinase inhibitors is, as I said in the beginning, I'll speak a little bit about it. Now, EG5 inhibitors or spindle kinase inhibitors, you know, everybody knows mitosis and meiosis, am I right? How the cell division takes place. Now, in the cell divisions, we have chromosomes, am I right? Like when the cell division takes place, the chromosomes have to be, they become haploid, am I right? Therefore, they are pulled apart and aligned. In that, there is one KSP, is a mototubule enzyme, EG5 enzyme, which basically tags along and carries the along the tubule, it segregates and aligns the chromosomes. So our proposal was to inhibit that chromosome. So in this stage we synthesized analogs of K858. K858. This is a K858, a very potent molecule, but nobody had co-crystallized K858 with the enzymes, and we were able to do it. And as you see, this is a racemic mixture, and we were able to see that the R isomer was actually active, the S isomer was not active. So this was our contribution towards this study. Going further here, anti-cancer agents, we work on some of the very good anti-cancer, we have worked extensively on that, on kinase and CDK kinase and spindle kinase inhibitors. Okay, I just go quickly, coming to this, as I said, we all academics, sometimes we do the bench work, but most of these people are the workhorse people, like they work in the lab, so I need to acknowledge them. So this was my group in 2015, 2017, 2018, and this is the UKZN campus, and this is the current group that I have from here. The CH activation work was done by Sachin Moite, and so these are my group people without whom this work wouldn't have been possible. All the ideas that is generated wouldn't have been to come to reality. My collaborators, these are my collaborators who have funded or have added to my science and helped us grow. So, come back again. Thank you.
Thank you.
So, this is my family, my wife Prithvi, my son Dheera, and my daughter Vivi, that's myself. So, we live in Durban, and this is St. Lucia, hippopotamus. And believe me, these are the baby hippopotamus, they're not the fully grown hippopotamus. And more people in the wildlife get killed by hippopotamus and bisons, not by the snake bites or the lions and all. So they're very aggressive animals. So just lazing around on the river bank, Indoloki river bank. So it's beautiful. If you come to South Africa, you'll experience all this wildlife. Most of the Discovery channels in National Geographic are either from South America or from Africa. So without these two continents, you wouldn't have most of the wildlife. Thank you very much once again for giving me this opportunity. For exceeding the time. Yes, bro.
Very nice work.
Thank you.
I have one question regarding this speech expression style.
Okay.
Right? You have used excess of style.
Yes.
What happened to
When we used the conversion was not very high and we were unable to get the desired results. But selectivity was still very appropriate. The major product was.
have you tried to observe what happens to the rest of that style?
No,
Any observation?
no, we haven't done that. We haven't. But I will.
Great work.
Okay, thank you, sir.
Thank you so much, sir. Now I request Professor S. C. Ahmed to facilitate Professor K. Rajshekar.
Thank you. Thank you, sir. Thank you.
Thank you so much for the talk, sir. So we have next speaker, Dr. Bakai Iliona, and she is from University of Debrecen, Hungary. Dr. Iliona is currently serving as an associate professor at the Department of Pharmaceutical Chemistry within the University of Debrecen since September 1st, 2022. Her academic journey began with graduating from the University of Debrecen as a certified chemist in 2004, followed by obtaining an English translation qualification in 2005. She further pursued her academic pursuits, earned her PhD degree in 2009, and completing her habilitation in 2021. Throughout her career, Dr. Iliona has contributed significantly to scientific literature with 47 articles published in reputable journals, garnering 266 independent citations, highlighting her notable research contributions and impact in the field. Warm welcome, ma'am. Over to you.
about glycopeptide antibiotic therapies against viruses. I came from Hungary, from the University of Debrecen. According to the World Health Organization, among the most important threats to the global health, we can find several viruses, for example, influenza, Ebola, dengue, and HIV. And there are not enough or no effective medicines or vaccines against these pathogens. And we can find antimicrobial resistance as well among these threats to the global health. The WHO has a list about the most important diseases, the priority diseases, which we have to deal with. These are COVID-19, Crimean Congo hemorrhagic fever, Ebola virus and Marburg virus disease, Lassa fever, Middle East respiratory syndrome, and severe acute respiratory syndrome, Nipah and helipaviral diseases, Rift Valley fever, Zika disease. This is caused by unknown pathogen, which can cause next pandemic or epidemic. For example, COVID-19 was a disease a few years ago. There are about 5,000 virus species that we described so far. Many of these are harmless, but others can be quite dangerous. And unfortunately, there are no effective medicines or vaccines against most of these viruses. And with the increase of the population on the Earth, the possibility of new epidemics and pandemics increases. Therefore, it is a very important goal for researchers to find more effective antiviral drugs. Besides viruses, the antimicrobial resistance is also a huge problem, and the most important problem is the resistance of bacteria. The antimicrobial resistance is responsible for about 33,000 deaths annually in the European Union, and 25,000 death cases occur as a result of infections caused by resistant bacteria. Globally, this number could be as high as 700,000, and until 2050, 10 million deaths are predicted yearly if current infection and resistance trends are not reversed. Resistance developed against some level of resistance developed against most of the known antibiotics. Therefore, it is a very important goal to find new and better antibiotics as well. Glycopeptide antibiotics are natural or semi-synthetic compounds that inhibit the cell wall synthesis of gram. You can see the structure of diabetes. Sorry. So here you can see the structure of typhotamine and vancomycin. They are the most known glycopeptide derivatives, but there are semi-synthetic derivatives as well in use, like gandolancin, pelalancin, and orisavancin. It is known from the literature that some of the glycopeptide antibiotics have antiviral activity as well. We started our work with ristoceptin. Ristoceptin is also a glycopeptide antibiotic, but it is not used as an antibiotic because of its platelet aggregation property. But ristoceptin aglycon derivatives have antiviral activity. We started our work with ristoceptin. We eliminated the sugar moieties from the molecule because these are responsible for the platelet aggregation property, and we got this aglycon which has antiviral property, and it retains the antibacterial properties as well. So we prepared several types of ristoceptin aglycon derivatives with activity against influenza strains, but ristoceptin can not be bought commercially because so it can not be buying higher amounts. That's why we started work with teicoplanin, which is, as I said, also a glycopeptide antibiotic, and it is known about teicoplanin that it has antiviral activity as well. So teicoplanin is a mixture because we can find several lipophilic chains connected to this sugar moiety, and it is very difficult to handle this molecule chemically. Purification of the product is not easy, so therefore we eliminated the sugar moieties from the molecule, and we got this teicoplanin pseudoaglycon derivative, which has no antiviral activity, unfortunately. But it has antibacterial activity as well. So we planned new derivatives to increase the antibacterial effect of teicoplanin pseudoaglycon, and we would like to increase the activity against resistant bacteria as well. We modified the N-terminal amino group of this molecule. For example, by the production of amides, or we can transform this amino group to azide, and we use azide link reaction for the derivatization of this amino group. So what is the mode of action of glycopeptide antibiotics? They inhibit the synthesis of the cell wall. The bacterial cell wall consists of sugar, oligosaccharide, or polysaccharide chains connected together with oligopeptide short chains. The glycopeptide antibiotic can connect to the end of the oligopeptide precursors, and they can hinder the transpeptidase enzyme, the function of the transpeptidase enzyme, and the transpeptidase enzyme cannot create these cross linkages. glycopeptide antibiotics connect to the D-alanine end of the pentapeptide precursors, and during the resistance, the structure of this pentapeptide precursor changed, and the strength of the connection between the glycopeptide antibiotic and the pentapeptide precursor will be decreased. So to increase the antibacterial effect of teicoplanin derivatives, we planned to prepare lipophilic molecules, lipophilic or amphiphilic molecules from glycopeptide antibiotics. So if we connect a lipophilic pair to a glycopeptide antibiotic, we get amphiphilic molecules. Amphiphilic molecules can create aggregates, and the aggregates can connect to the bacterial cell wall or the bacterial membrane, bacterial cell wall stronger because of this multivalent effect. So therefore, we plan to prepare glycopeptide antibiotic derivatives with lipophilic chains. So this was our first synthesis when we prepared teicoplanin pseudoaglycon derivative equipped with a sugar moiety which has two octyl side chains, and we sent these two molecules for antiviral and antibacterial test as usual, and unfortunately these derivatives had just weak antiviral effect, antibacterial effect, and it was a serendipity that these derivatives were highly active against influenza strains. They have a higher or same effect than zanamivir, which is a medicine used against influenza. And it was shown that these derivatives hinder the attachment of the virus to the host cells. After these derivatives, we prepared several types of derivatives with lipophilic chains, but the antiviral activities was not as high as in the case of the original dioctyl derivatives, and these derivatives had high cytotoxicity because of the long lipophilic chains. Therefore, we planned some new derivatives. We knew from our previous experiments that high lipophilicity of the side chains is essential for the antiviral activity of the glycopeptide antibiotics, and unfortunately high lipophilicity usually goes hand in hand with high cytotoxicity. But cytotoxicity can be reduced by hydrophilic linkers. For example, if we use a hydrophilic ethylene glycol linker between the lipophilic tail and the glycopeptide antibiotic. So after this knowledge, we planned new derivatives with perfluoroalkylated side chains. Perfluoroalkylated chains have hydrophobic and lipophobic property in the same type, but they have no membrane activity, and therefore we assume that these derivatives will have no cytotoxicity. So here you can see that the triple derivatives from vancomycin and tacrolimus and icon with perfluorobutyl and perfluorooctyl side chains. These tacrolimus pseudoacenicon derivatives with perfluorooctyl and perfluorobutyl side chains have very good antiviral activity. They were highly active against influenza strains, and they have no or low cytotoxicity. As you can see, the mother molecule, the tacrolimus, has no activity against influenza viruses. So during our work, the SARS-CoV-2 pandemic began its journey around the world. So we started our work against SARS-CoV-2 as well. The spike protein of SARS-CoV-2 has a furin 3D site. Furin is a human enzyme which can activate the spike protein. And the furin enzyme can connect to an arginine-rich region, and arginine has a guanidinol group. Therefore, we plan to prepare guanidinol derivatives from tacrolimus pseudoacenicon to hinder the function of furin enzyme. To understand the mode of action of our derivatives, I would like to show you the replication cycle of the SARS-CoV-2. After furin activation, the virus can attach to the host cell at ACE2 receptors, and TMPRSS2 enzyme can activate the spike protein, and the fusion occurs with the cell. Or TMPRSS2 enzyme is expressed in respiratory cells, but if the cells have no TMPRSS2, the virus can get into the cell by endocytosis as well. And in this case, the cathepsin enzymes can activate the spike protein, and the genome can get into the cell. During the replication, polyprotein produces, and the 3CL enzyme can cleave this polyprotein to non-structural proteins. So this is a very complex process, and we can inhibit this process in many points. Our colleagues investigated the antiviral, the anti-SARS-CoV-2 activity of our molecules. They investigated our molecules in viral E6 cells. This is a green monkey kidney cell line which has no TMPRSS2. So the virus can get into the cell by endocytosis, and cathepsin enzymes can activate the spike protein. So in this case, except perfluorobutyl derivative, the perfluoro derivatives and the originally synthesized octyl derivative and the monoguanidine derivative have high activity against these the SARS-CoV-2 in viral E6 cells, and tacrolimus itself has also activity against SARS-CoV-2, and these derivatives were not cytotoxic in viral E6 cells. CAL3 cells are lung epithelial cells. and they express TNPR-SS2. In this case, just two derivatives were active, the diethyl derivative and the perfluorooctyl derivative. And these derivatives were active against the human 229E type of coronavirus. So our colleagues investigated the possible mode of action of these molecules. As you can see, these derivatives have just weak inhibition property against cathepsin L enzyme, and the triguanine derivative derivative high inhibited this cathepsin L enzyme. There were just very weak inhibition against C3-APRO enzyme, so the viral C3-APRO enzyme. And these derivatives just weakly inhibit the ACE2 spike interaction. So we couldn't investigate all the possible enzyme inhibition or other mechanisms. So we suppose that other enzymes are also involved in the mechanism. So the exact mode of action is not known yet. We investigated, or our colleagues investigated. Okay. Our partners investigated the antibacterial effect of these molecules, and as you can see, the monoguanidine derivative were very active against these nosocomial strains. It has similar activities than the best glycopeptide antibiotic, the orithomycin. And they investigated the activities against nosocomial teicoplanin and vancomycin resistance strains as well. And as you can see, this monoguanidine derivative very active against these resistant strains. It has similar or better activities than the best glycopeptide antibiotic orithomycin. So I have to finish my lecture. So the and this
and the other structural part, if you look at the isolation ring, they are embedded within a protein core, which is the aproprotein matrix, right? And all these functionalities, all these functions are fine-tuned by several non-covalent interactions which prevails within this aproprotein matrix, through the aproprotein matrix. And nature somehow fine-tuned this property, otherwise it should have been a single property, not for a diverse application, but for a single particular application, isn't it? So how does nature does it? Can we do it? Can we biomimic that system for practical application, for our applications? And our group has been working in a couple of different aspects. One, not to generate the exact biomimetic catalysis, but try to understand how exactly does this non-polar interactions work. Can we stabilize the key intermediate? It's not visible here, but there's a hydroperoxy species, which is a highly unstable intermediate transitory form in the reaction system. Can we stabilize it with a small peptide chain, or maybe with a metal center around it? Can we utilize this redox active molecule, connect them together in precisely in a one-dimensional layer-like structure, form one-dimensional thread-like structure? Can we pattern it onto a surface for molecular wire-like applications? Or we are also trying to understand the self-assembled behavior of all these moieties, so that we can generate different kind of materials for different applications. For today's talk, to make it more connected with the integrated chemistry, biology, or the translational medicine part, I thought I would be talking more about our efforts with this bioluminescence behavior. Can we use this bioluminescence behavior of these molecules for bioimaging or any further applications? So I will just try to give you two examples in this particular domain. Okay, I will ignore this part where I was trying to make comparison. Okay, I will ignore this part where I was just trying to make comparison or trying to understand why we want to target selectively mitochondria, because it's a redox active center, lots of activities are happening within this mitochondria. And just to highlight one key aspect of the mitotractors which has been available in say commercially available or has been frequently reported in the natural literature part, almost all of them, I'm saying almost, not exactly all of them, some of them are neutral species, but almost all of them are actually the cationic species. They have some kind of cationic charge residing on that molecular structure, right? The problem with this cationic species is it can rupture or it can play around the membrane potential of this mitochondria. That means you will not get an exact picture of how your mitochondria is working, okay? They have a couple of other issues as well. I will not go again detail in that. But so the idea was, can we have a simple analog which can go reside inside the mitochondria and gives you the exact picture? And one aspect which I missed with you, I was trying to compare the green fluorescent protein versus this flamin-based fluorescent proteins, is that this green fluorescent protein fails to give you any significant fluorescent emission in case of hypoxia condition. That means inside the cell, if the oxygen concentration is less, that's the hypoxia condition which prevails in certain disease state. So can we monitor that? We cannot monitor that with these analogs and so on. So what we have tried to develop is a small simple molecule, if we can. What we have tried to do is, ignoring how we have designed this, that was the previous slide, how we exactly came up with this structure. So we have tried to just connect this TPA analogs connected with the simple
Before I start, not get molecule like this, and doing so it make the two carbon nitrogen bonds, and this reaction is highly efficient. And you see again, this molecule is heterocyclic molecule, highly nitrogen rich molecule, conjugated with this sugar moiety which is called 2,3-farmyl glycal, either micro condition or thermal condition, and gives this molecule. And this work is already been, very. This is in ASAP in GOC. And another story which I will be sharing with you, where we have conjugated this 2-hydroxy naphthoquinone, which is again a natural product molecule, with this same precursor, 2C chemistry journal, and in ethanol. In 5 minutes, we are able to make this a skeleton. This is our proposed. 2C-farmyl glycal, they are chiral synthons, and they can successfully couple with 1,3-dinucleophiles, and giving the this pyrrolopyrimidines or this pyranopyrones skeleton. And there is a very recently paper published from the Prasad-farmyl glycal, rather 1C-farmyl glycals, and coupled with coumarins and give this this kind of skeleton. From withdrawing group or donating group, isopropyl, methoxy, and even in bicyclic compounds, all these beta ketone were very good. And then after having these beta ketone in hand, the next step was to couple this beta ketone with thiourea, and which we have done in the presence of derivative. And this was then treated with the methyl iodides. So this SME was formed, followed by the hydrogen iodide treatment. Nitrogen containing compound, and coupled with this 2C-pharma. Whereas in ethanol at 80 degrees centigrade thermal condition, in 6 hours it was a 54% yield, but if you elongated the reaction time, the yield would also reach up to 70-75%. So after having the optimized condition in hand, all diverse precursors having the withdrawing donating group, different stereochemistry, but this mechanism was not that much difficult, so that's simply like if the 2C-pharma, what we proposed, this is our proposed mechanism. 2C-pharma proton makes this iminium and then phosphorate to this particular carbon, because this is oxonium, which is electron trace, make this another cycle, and then followed by ring openings gives this particular product, 32. And then we have done some molecular docking. The score for selected molecules was quite high, which is minus 10.56 kilocalorie per mole, and also we have done this one. This is our molecule, and the red part is the one natural ligand for this. So with this work has been published in this JOC. The next story where we have the precursor, this where we is you see these are a few of the natural protein molecule, and even this vitamin K, it has the nepthoquinone unit in it, and this is another natural product. This is like doxorubicin, which I was talking about. It has the nepthoquinone units and tetracyclic skeleton. And inspired from these natural products and along with some medicinal medicine, we have designed this molecule, which have the chiral center as well, and having the nepthoquinone unit at the same time. So this is our plan for making these molecules. We plan to take this 2-hydroxy nepthoquinone, which is commercially available, and only one. So this is 1,3-dinucleotide. And if you see this precursor, this is electrophilic in nature, and this carbon is electrophilic in nature. So like bringing 1,3-dinucleophile close to 1,3-dielectrophile, and to see whether do the coupling or not. So the precursor needed for this one was this one and this one. This one synthesized again from the 2C-pharma glycol during the Wills-Murray reaction by reported method by Professor Subramaniam, reported long back in 1991 at IIT Madras, and we use the same precursor. We plan to subject this the 2-hydroxynepthoquinone. As I mentioned, only one precursor, which is number 9, it is commercially available. Rest we have to synthesize. So again, starting from the beta ketoester, by using auxiliary chlorides and like Friedel-Graf acylation, the reaction, and followed by the sodium hydroxide treatment, so this can lead to the decarboxylation, and finally gives this product. These 6 precursors we have inside in our laboratory, and then it was a spin for the transformation of into our design product, and we identified number 5 here, where the pyridine was used as a base in 0.1 equivalent, and toluene and acetic acid in 9:1 ratio, and at 80 degrees centigrade, within 20 minutes we were able to make this molecule in 80% accelerated. And then after having optimized condition in our hand, the rest of the 2-hydroxynepthoquinone was successfully coupled with this 2C-pharma glucal as well as glycal, means the green highlight part here, you see there is a difference in the stereochemistry, and this precursor was prepared. And this is the proposed mechanism again for this transformation. This 2-hydroxynepthoquinone attached to this position makes this intermediate, which followed by the beta elimination gives the one like the hexatriene, 1-octatriene system, where we have 3 double bonds, each undergo 6 pi electron electrocyclation and finds our design product. And in this synthesis, while this product, and again this ASI has done some docking studies for these molecules. Because we haven't done the screening of these molecules because of lack of biological facilities or identified laboratory, which we will be doing soon. So we have done the docking studies against one particular protein, and we found these molecules, they have good binding score in the again in the small story. That is, we have developed a method glycal with alpha known derivatives. And why these molecules again? These molecules are present in many natural products. If you see this one, this is a few of them are drug molecules having different biological activity. So we start from this, and even these skeleton here, if you see, highly nitrogenous attached with the sugar, and they have drug molecules. So inspired from this, we have designed molecules something like this here, which is shown in this particular slide, and which having this pyrimidinone skeleton fused with imidazole, pyrimidinone, and coupled with this sugar part here. Here sugar is in acyclic form. So then we have searched the literature. We found that this alpha enone derivative, particular paper published in Organic Letter, where this alpha iodinones by different kinds of metal has been used for doing a lot of alkylation and arylation at this point, at this particular site. Later on, a paper published in this was from Chinese group in 2016, where they used this particular derivatives about the similar kind of work which we have done, not exactly the same, but they have made this imidazole kind of derivatives by using these precursors. So this is our work. We use the same starting material, alpha iodinones, and coupled with this pyrimidinones, and we got this particular product. And this is published in European Journal of Organic Chemistry. So starting from beta ketoesters, coupling with this guanidine derivatives in the presence of base, successfully give this product, which is pyrimidinone derivative, and all 9 precursors have been synthesized, having the electron withdrawing and donating group at the phenyl ring in very good yield. And then these alpha iodinone derivatives, this was also prepared in the laboratory, and they were subjected for the coupling reaction by using different ways, by using potassium carbonate, potassium tertiary butoxide, and then sodium carbonate. Entry number 12 here, by using potassium carbonate around 3 equivalent in GMSO at room temperature. Within 4 hours, we were able to make this compound in 8% acidic yield. And this piece of work is detailed study in McMillan has been published this year in GGOC. And these are we took the substrate scope for this transformation. So by using different substrates here in R1 and R2 position, you see all sort of things we have tried, like cyclic, and then CF3, bromo, fluoro, isopropyl, methyl, methoxy, and they were all transferred into successfully into the final product without having any problem. And this is purification, because if you see this one, this was derived from the glucose, and then we thought doing the same reaction with the galactose derived one, and it worked perfectly. We wanted to introduce the stereochemical diversity into this alkyl side chain. And this is the proposed mechanism for this particular transformation. We propose like this, this amine group here of this moiety attached to this position, which is electrophilic in nature. So, and makes this substrate here, then followed by attack of this double bond onto this iodine, and then this ultimately leaves the cyclic compounds, and followed by this ring got open and gives this product, and which is equilibrating with this final product. Pyrazoles and imidazole triazoles by using these alpha iodinone derivatives. And why alpha iodinone derivatives? Because imidazole, pyrazoles, and imidazole triazoles skeleton, they are present again in many drugs, currently used drugs, which having this skeleton, along with this acyclic polyol side chain, which is present into like this riboflavin molecule. And this is a molecule which is reported by a Korean group. They mentioned that this molecule is an anti-malarial molecule. So you see this acyclic polyol side chain, which having predefined calcium. And those of you from synthetic background, you might be knowing, bringing the stereoselectivity into the acyclic chain when you do chemical synthesis, when you try to generate the chiral center, is very, very challenging. We can inherit this from the nature. So, and this is how we have proposed this molecule, our design molecule, and for this, we plan to use this alpha iodinone derivatives, which has 2 predefined chiral centers, which are coming from the substrate, from the nature, and coupling this with 3 amino pyrazoles or 3 amino triazoles under suitable condition to get the final product. Very simple approach. And so this alpha derivative, this was synthesized starting from the glycols by oxidation followed by the selective hydrogenation, and which is a reported method. And then after having these two substrates in our hand, we did the reaction optimization, different ways, different stone volume at elevated temperature 90 degree, within 2 hours we get this product which is 78% isolated. And we also did this reaction with microwave. That's very obvious. This reaction was finishing in 10 minutes rather than 2 hours and giving almost 80% isolated. So then we looked the substrate scope for this transformation. So this alpha derivative was successfully coupled with the differently substituted 3 amino pyrazoles, and like the library of compounds were synthesized, and this code we support this for this last level synthesis. For this molecule, and we found our molecule, this was SCK, which is protein, this is tyrosine kinase protein, and this molecule matches the same site. So might be these molecules is acting the same way, inhibiting the tyrosine kinase agent. Docking score for this one around minus 18 kilocalories per mole. So with this, I would like to conclude that can be metal free by simply like using the pyridine or potassium carbonate as a base, but you identify the suitable substrate. You bring two substrates which like each other, so they make the compounds. And these molecules, the heterocyclic molecule which have been generated, they are highly privileged scaffolds. They are present in nature, like imidazopyrazoles or imidazopyrazoles, increases the solubility, and also it improves the selectivity towards the tumor cell because of the Barber effect. So with this, I would like to conclude. Here I would like to acknowledge like the JNU and BHU because we have used their lab facilities. But as I mentioned in the beginning, this whole work was self-funded, and the work which I have published in GOC was done by Ghanshyam, and this eGOC work was by Vinay, and Ashish Khanna has done the docking, molecular docking, as well as contributed into the C paper. And so in like 30 seconds, I would like to just showcase my laboratory which we have recently developed at the JNU, because as sir mentioned, I moved from the BHU to JNU. So this is my laboratory, and this was again developed on the PPP model, which is a model proposed by our Prime Minister Modi, and this was funded by Mr. San Sangaria. I should acknowledge him as well, because when I joined, I got the empty space, and this all furniture actually provided is purchased by Indian company that is Citizen, and this is the first batch of chemical when I arrived in my laboratory. I was quite happy. This is my son. He is doing graduation at Delhi University, and then this is Mark from Germany who was visiting, and Kavita is here. She is presenting poster today. This is Radhit Tyagi and Saurabh is standing here, and this is my first batch of a student, let's say, when I started my career at JNU, and they work hard, so that's why I'm today here. And this is the first rotor we got from the from Indian made again. This is working perfectly fine as the head off. This is I think copied from head off. Okay. And this lab was inaugurated by then Vice Chancellor, Jagdish Kumar. Then later on, our first woman Vice Chancellor to the Jawaharlal Nehru University, Professor Pandit, he also visited the lab, and he was then Dean, Gaddafi, and then many visitors actually came to visit this lab, and many people, and this is the how the School of Physical Science looks like in the front, and these are my students. This is my actually grand batch of my student at the JNU, and this is one of the statues which has been inaugurated by our Honorable Prime Minister on birth anniversary of Swami Vivekananda on 12th November 2020. So this is now become selfie point in JNU. So we have to take a picture there. And with this, thank you all for the kind attention.
We are running short of behind schedule. Can we? Yes.
Yeah. Thank you sir for nice presentation. In optimization table, write many mixture of solvent that water and THF like ethanol. Then what is the change in properties of the solvent?
No, so let's say when you take the THF, so THF is relatively less polar solvent. So when you add ethanol to it, So ethanol does two things. It increases the polarity and also it provides the protons. Because if you see the mechanism, there is one proton is needed.
From ethanol?
Yes, from ethanol.
Any other question? Anybody?
Sir, one question from us.
Yes, please. So, and actually I run a little fast because we want to go to for sightseeing. So I request to another next speaker to finish quickly.
That's great, yeah?
Can I ask or not, sir?
No, you can ask.
No, you can ask to him, but his advice is from the expert.
So my small question is that have you optimized about the green matrix of the reactions? We have checked the atomic economy and all those parameters.
Yeah, as far as atom economy is concerned, because there are coupling reactions. So there is no loss, only loss of whether it's H2O molecule or HI molecule, something like that. So in terms of atom economy, yes, but I would not claim this is totally green chemistry because I use some of the DMSO as well. But wherever I use the ethanol as a solvent, potassium carbonate as a base, that you can consider as a green reaction. Yeah.
Thank you so much, sir. Now I request Professor P.K. Chaudhary to facilitate Professor Ram Savitha.
Next speaker is Dr. Dhruv Kumar. He is Professor and Cluster Head, School of Health Sciences and Technology. He is among the top 2% scientists in the world in the field of cancer biology and oncology. His brief introduction to him, he is from USA. So I'll just ask Dr. Dhruv Kumar to start with the presentation.
Okay, great. So thank you very much, sir, for a kind introduction. And before I start, I would like to thank my dear friend. Today I'm going to talk about combinatorial therapeutic approach. But what are the components which are there inside the solid tumor? That's very difficult to understand, and for the common people. And once you know so. It is moving now. So, when you see the geographical distribution of cancer in India, in the most of the cigar. In Rajasthan, they use cigar. These things, and as Dr. Verma mentioned this morning, that around 4,000 chemicals are there. So what it does actually? So it does this. One of the important factor for this is the cancer when you use tobacco product. Next. Yeah. So coming to the head and neck cancer, there is statistics actually. In India, it's a topmost cancer in men. Okay, but the growth of head and neck cancer in women is also increasing. Why? The recent trend that you see in India, women have also started taking tobacco products. And this is historically in India, we have not such kind of system. So that tobacco related cancer is now increasing in women as well. But in men it's a topmost cancer. So, but you see, this morning my dear friend has talked about this morning, other chemist Dr. Rakesh and Dr. Misra, he talked about the chemical composition, chemical formulation and all. But this formulation gets vetted, validated when you do the animal work or cell culture work, then it goes to the clinical trial level, then it comes to the clinical practice. So it is very, very important for all of us what kind of research we are doing and how we are going to translate from bench to bedside. So our lab is mainly working in that approach, how we translate that molecule, those hard worker chemists they are synthesizing day and night, we can pick from there and to test in the lab and to further to propose it for clinical trial. But you see, there are lots of diagnostic tools are available. Lots of therapeutic approaches are available. Still, the survival rate is very, very less. And what is the reason? I will discuss briefly about all these things in my next slide. As you can see here, when you see cancer patient, cancer does not grow in one day. It's not a viral infection, bacterial infection. Something takes years and years to grow inside our body. And then it becomes, the cancer. So if somebody is having, unusual changes in their body, some kind of, the patches that you observe, immediately one should go to the clinician, immediately one should go to the doctor and they should see. Because as you can see here tongue, the difference is very minimal, very little difference as you can see. Sometimes, our lymph node gets swollen and that we ignore such kind of, the things in our body. But it is very, very important to examine our body by ourselves first. Okay, what kind of changes are happening? And if you see any kind of changes are happening there, then you please go and consult the doctor. The right therapeutic approach. So in our lab, we are working on autophagy and metabolic aspects of cancer with respect to the cancer cells and tumor microenvironment. CV Raj, my PhD student, he has just completed her PhD. Vaisali, she has completed PhD in 2022. She was working on HPV related oral cancer. Let me tell you, not only tobacco product, there are several other factors. Somebody mentioned about 4NQ, I don't know, in this morning lecture. and there are chemicals that can induce tumor actually on tongue in oral cavity. So if you see, together with that, there is a virus called human papilloma virus, HPV. So HPV is associated with the cervix cancer, mainly 99% cervix cancer is associated with HPV, but there is a cancer called oropharyngeal cancer that is associated with HPV only. So Vasali was working on it. And oropharyngeal cancer, the cervix cancer can be prevented well through the vaccination. Vaccines are available now. One can, below the 25 years of age, the girls can take that vaccine to have these preventive measures. As well as mutational analysis. Then the second part that we do, that we study, we isolate cells from the tumor, solid tumor, that we call primary cells, and mainly the tumor associated fibroblast cells, and then we study the crosstalk between cancer cells and tumor associated fibroblast cells. So the interesting thing that just now the previous speaker has mentioned about the metabolism briefly, and the metabolic related chemicals. PET CT scan. And what is PET CT scan? Is that there is a molecule called 2-deoxyglucose. It's a modified form of glucose where you have a fluorine tag, and that's why you inject those molecules to the patient, and at the second level of glucose metabolism, when fructose gets glucose 6-phosphate gets converted into fructose 6-phosphate, this molecule gets trapped into the cell. The further metabolism gets hampered, stopped there, and that's why through PET CT scan you would be able to see the spot as a cancer cells. So these are actually metabolically active cells which are present in solid tumor that you detect through PET CT scan. So this is very important part that actually a chemist actually helps a biologist or the clinicians to work on the therapeutic development as well as diagnostic part. Now, I cannot move ahead if I miss this thing, this slide. The great scientist of the century, I would say that it's Otto Warburg, and on his name we have given this concept, the Warburg effect, that Dr. Misra has mentioned just now. And why? Because cells, they perform aerobic glycolysis, and that is the cancer cell. I will discuss briefly about that. My colleague, Dr. Asutosh, he has mentioned about the mitochondrial tracker. There are lots of things that you can do with the metabolic switches in cancer cells. So he got Nobel Prize. He's a German scientist. He got Nobel Prize long back, and he gave that concept that cancer cells utilizes the glycolysis even in the presence of oxygen. Nobody actually looked for that because sometimes when you invent something, some invention goes into the the dustbin. Even in the Nobel Prize, something related to that. the clinicians, they treat cancer patients through restricting just their metabolic activities. They will stop, they don't give anything which has a glucose molecule. And that's why means integrated therapy works well now. If Ayurveda, homeopathy, allopathy, when they put all together, it works. Now, coming forward, okay, just one thing I wanted to mention here. So what happens in normal cells, this mechanism works well, this mitochondria, but in cancer cells, this mechanism works actually. Pyruvate gets converted into lactate. Through lactate dehydrogenase A enzymes. And then this lactate goes out from the cell because the pH of this lactic acid is less, it's acidic. If it stays inside the cell, then it automatically cancer cells will die. And this is the reason why MCT1 expression goes up and it exports lactic acid from here. So in this lecture, I will talk about how we have inhibited. So we have done some bioinformatics analysis to understand the gene expression and all. There is a receptor tyrosine kinase, c-MET protein, and metabolic related enzymes are there, hexokinase 2, LDH, MCT1, as well as this GLUT1 expression. And we saw there is a positive correlation with all these cancer associated genes as well as the metabolic related genes. This is the cancer associated that we call receptor tyrosine kinase. Now, further we have done bioinformatics analysis to understand their network, whether they are associated with each other or not. Then we have done the correlation analysis with receptor tyrosine kinase as well as these enzymes hexokinase 2, phosphoglucokinase, LDH, and MCT1. So this is very important thing that in the beginning of my lecture I mentioned. If you identify cancer patient in the early stage, you would be able to actually treat them well, and cancer patients can survive normal life. This is the reason what that you see here. In the first stage, this is the stage-wise analysis of gene expression. In the first stage of cancer, you see, even the genomic alteration gene expression is less normal. Those are the driver genes that leads to the cancer progression. The way when it grows to the stage 2, stage 3, stage 4, the expression goes up. So, therapeutical point of view, preventive point of view, early stage works really well. Okay. So further we did a correlational analysis to understand the immune functions. These are the real patient data. When we do the NGS analysis, we put our data on a data set that we call GEO data on NCBI. So anyone can download those data, can do the reanalysis and find out the correlation. So as you can see here, all immune cells are negatively correlated. It means when cancer grows, your immunity goes down. But interestingly, if you can see here, cancer-associated fibroblast cells, the number of cells are increasing when cancer grows. So there is a positive correlation with cancer growth and cancer-associated fibroblast cell. So what we did, so just to make you understand, in an easy way, I have prepared this schematic. So as you can see here, these are carcinogens, viruses, diet, or smoking, whatever. And this is your tissue system. This is your epithelial cells in your tissues. So when our cells get exposed to these chemicals, what happens? There is a change in called that happens through the mutation. Our DNA gets mutated. But the beautiful thing is that naturally our cells repair those things. That is called repairing process. You don't have to take medicine or anything for that. Our cells gets repaired because our body has such kind of potential. Okay, but when you give these things continuously to the cells, when you bombard with the chemicals and all, then after sometimes your cells, the tissue system recognize that as a normal cells, and then they allow them to actually pass this checkpoint of cell cycle. In the morning session, someone was talking about, I think Dr. Raj was talking about this cell cycle checkpoint inhibitor. Okay, and then single cell grows in a group, and that is called the tumor. This is a group of cells actually that you see here. And these cells, then what what these cells they do inside the tissue, these cells then secrete some of the growth factors, protein, small protein, peptides inside the tissue microenvironment, and then it transforms this, the normal fibroblast to the cancerous. That we call cancer-associated fibroblast. Then again, what these cells they do, cancer-associated fibroblast cells secrete some. These cancer cells and these cancer-associated fibroblasts, they help each other. There's a mutual crosstalk. Okay, and this is the reason why when we identify this mechanism, we have blocked this crosstalk through the inhibitors, where chemist has synthesized even the specific inhibitor for the molecule. So, if you see the solid tumor normally you have multiple cells, not only cancer cells. These are the cancer cells, macrophages, immune cells, blood vessels, and in the background that you see, these are the fibroblast cells. So if you target just cancer cells, you will not be able to kill the, reduce the tumor burden. So you will have to, use the strategy how combinatorial therapeutic approach you are going to use for, to kill the cancer cells together with the other cells. Then this is the patient, the sample, the histological sample. As you can see here, this is the cancerous region, and this is the region that is associated with tumor-associated fibroblast. Now, this is very, very interesting to understand why we should use this combinatorial therapeutic approach for cancer cells as well as cancer-associated fibroblast cells. So, the level of this cancer-associated fibroblast cells, if you see, in oral tumor and specifically in the head and neck region, in solid tumor and breast tumor, it is really very high, very high. So if you are killing these cells together with cancer cells, you would be able to reduce the cancer burden at maximum. Okay. So we further went and we isolated condition media from the cells that we have. This is the mechanism actually that we follow. This is the cells isolated from solid tumor primary cells we call. We collect condition media, then we treat cancer cells. We measure cell proliferation, migration, invasion, and all other studies that we want. So here I would like to acknowledge our collaborator, Dr. Parvesh Mehra from Delhi Art and Medical College, who has provided us this oral tumor. We have isolated cells out of that, where you can see here the other cancer-associated cells isolated from the tumor in our lab. Then this is the mechanism in the beginning that I have discussed, that these two cell types, they help each other in tumor growth. So our approach was to block this activity and block this activity to stop the tumor growth. So some of the experimental data I'm going to share here. Glycolysis is very high in cancer cells. Theoretically it happens very high. But mitochondrial aspiration in tumor-associated fibroblast cells are here, as you can see. These are two different cell types. In one cell type, cancer, we have more glycolysis. In TAP, we have more mitochondrial aspiration. So this is the foundation, this is the basis of this study from where we started actually. Then we have blocked glycolysis in cancer cells, mitochondrial aspiration here in the tumor-associated fibroblast cells to reduce the tumor burden. Then further experiment we did with the cell lines, and we saw the increase in metabolic-related enzymes. All here we have measured lactate dehydrogenase A. If you remember, in the beginning I said that lactate dehydrogenase A is a very, very important enzyme that converts pyruvate to the lactate. So lactate activities with glycolysis